Good morning, everyone. I'm Katherine Schaefer, reporter at GenomeWeb, and I'll be your moderator today. The title of today's webinar is Spatial AI Biomarkers, Expanding the Eligible Patient Pool with Novel Histopathology Signatures, and it's sponsored by Nucleai. Our speakers are Justin Lucas, Head of Translational Research at Aldai Nortye, and Ken Bloom, Head of Pathology at Nucleai. You may type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar presentation. If you look to the bottom tray of your window, there are a series of widgets to enhance your webinar experience. With that, I'll turn it over to Ken Bloom.
Thank you for the introduction and welcome to this webinar. I am Ken Bloom. I head pathology at Nucleai. I'm a pathologist with more than 35 years experience as a surgical and molecular pathologist, and I'm going to give you a brief introduction to digital pathology and spatial biology and how the two have been combined with machine learning to provide new insights, improve and accelerate drug development, and ultimately improve patient care. So how many of you guys here remember the Beverly Hillbillies? Probably date myself a little bit, but it started out, let me tell you a story about a man named Jed. Well, I'm going to tell you a story about a guy named Ken, which parallels the evolution of digital pathology and AI. So back in 1981, I was a second-year medical student at Rush University in Chicago when I met Ron Weinstein, who was the chairman of the Department of Pathology. Ron was convinced that computers and emerging information technologies would change the practice of medicine, and more specifically, pathology. Because of my background in programming, Ron convinced me to do a post-sophomore year fellowship in pathology, and that decision changed my life. Ron was the director of the Central Pathology Laboratory for the National Bladder Cancer Project, funded by the National Cancer Institute, NCI. The lab reevaluated surgical and cytopathology diagnoses of patients who were enrolled in NCI bladder cancer trials before the outcomes of those trials were published. What they discovered was that the inter-observer variability was identifying dozens of treated bladder cancer patients that actually didn't meet the eligibility criteria for the trial. You can see this from the graph on the right, and some of the institutions had major discrepancy rates of close to 20%. Several unsuccessful attempts were made to correct the discrepancies, but none of them proved to be sustainable. My job was to design the database and the user interface for the Central Pathology Laboratory, analyze the discrepancies between the pathologists looking for trends that could someday be corrected. So even before I started my pathology residency, I was well acquainted with the problems of reproducibility in pathology. And that didn't even take into account issues that I learned later on, like pre-analytic variation and tumor heterogeneity, which also have a significant effect on reproducibility. It was these events that triggered the idea of telepathology as a way to overcome interpathologist reproducibility in real time. Fast forward a few years, and Ron recruits me to be a pathology resident at Rush. Ron had been conceiving the idea of telepathology, and together we designed the first system using a newly developed robotic microscope from Olympus, a customized robotic stage, a high-definition camera, first of its kind from Ikegami, and a satellite on loan from the Department of Defense. I programmed that first robotic system, but more importantly, I had the ability to review all of the cases I saw during my residency on both my microscope as well as a high-resolution monitor. It quickly became clear to me that I could diagnose the slides as easily on the high-definition monitor as under the microscope. We published a series of papers describing the system and how it could be utilized to network pathology services. I should also point out that one of my first published papers as a resident was a current topics paper for human pathology in 1985. It described the emerging field of artificial intelligence and how it might be utilized in pathology and medicine in general. The challenges that I put forth 40 years ago are largely still unmet. The bottom image shows a young Ron Weinstein and myself on the left at the first International Telemedicine Conference in Tromsø, Norway in the early 1900s. Throughout my career, I had the privilege of participating in the evolution of pathology and the emergence of precision medicine. As a medical student and resident, I was taught that medicine was an art and would always be an art because of the great variability in individuals and their tumors. But over the next 3 decades, I participated in the scientific advancements that reshape pathology and medicine. Things like immunohistochemistry, flow cytometry, in situ hybridization, polymerase chain reaction, next generation sequencing allowed a deeper understanding and more granular classification of disease. Understanding the hallmarks of cancer allowed us to identify and target vulnerabilities in individual cancers, and this led to the concept of precision medicine. Rather than treating every cancer patient with the same therapy and realizing benefit in only a small subset, the idea of precision medicine was to identify different subsets of patients likely to respond or not respond to specific therapies. This is accomplished through the use of a diagnostic assay. Most diagnostic assays assess a single biomarker, which is used to either select or deselect a patient for therapy. Unfortunately, most biomarkers have limitations, so we're constantly looking for new and better biomarkers. The first approved companion diagnostic test was the Hercept test from Dako, which is now Agilent. Hercept test was an immunohistochemical assay that measured the amount of HER2 protein expressed on the surface of invasive breast cancer cells. Patients showing 3+ overexpression of HER2 were deemed eligible for trastuzumab therapy. Unfortunately, interpathologist variability in the interpretation of HER2 turned out to be worse than the variability we saw 15 years earlier in the Central Pathology Laboratory of the National Bladder Cancer Project. This chart is from a study that I performed on behalf of Chromavision, one of the first digital pathology companies. 10 pathologists reviewed the same 100 immunostained HER2 slides and scored them as 0, 1+, 2+, or 3+, according to the guidelines in the package insert of Hercept test. Each case was called positive or negative based on the number of copies of the HER2 gene as determined by a fluorescence in situ hybridization assay. It is worth noting that the pathologists ranged in experience from experts to novices, but some of the worst correlative scores were among the so-called experts. Chromavision built the first whole slide imaging system that they called ACES, or Automated Cellular Imaging System. A HER2 application was developed to aid pathologists in the interpretation of the HER2 biomarker. The system created a 10x whole slide image of a HER2 immunostained slide and then measured the intensity of the brown stain in selected areas identified by the pathologist. As you can see from the bar graph, all of the pathologists performed better with the aid of image analysis, but even more importantly, they all performed with a similar high level of concordance. So even 25 years ago, we recognized that image analysis tools could aid the pathologist and democratize slide interpretation. Over the last 25 years, therapies have advanced, as has the need for improved biomarkers. The detection of molecular alterations or overexpression of specific proteins was the mainstay biomarkers for targeted therapies. But these type of biomarkers fall short in predicting response to newer therapies like immunotherapy or antibody-drug conjugate therapy. In immunotherapy, we attempt to re-engage the patient's immune system to recognize and kill their tumor. This requires an understanding of the patient's tumor, the tumor microenvironment, and the patient's immune system. Antibody-drug conjugates are therapies that have 3 basic components: an antibody or combination of antibodies that aid in the targeting of the drug to the tumor, a payload to kill the tumor, and a linker that releases the payload only when the drug is bound to its target. The payload can be a microdose of chemotherapy, a radioactive emitter, or an immune engager. The hope is that deep spatial insights will generate better companion diagnostic biomarkers for these new therapies. Spatial analysis combined with deep learning can assess histologic sections to predict molecular alterations, such as mutational burden, aid in the accurate detection and location of protein biomarkers like PD-L1, provide insight into the relationship between cells like lymphocytes and tumor cells, aid in the calculation of densities, fractions, and ratios, and uncover cellular neighborhoods not easily recognized with the human eyes. For antibody drug conjugates and multispecifics, deep learning can aid in the reproducible and accurate detection of target-expressing cells, especially at low levels of expression, assess heterogeneity of those cells, uncover the relationship and proximity between target-expressing and non-expressing tumor cells to better understand the so-called bystander effect, which is the ability to kill non-expressing tumor cells by a payload that is delivered to target-expressing tumor cells. While the concept of pathology-aided interpretation and the use of artificial intelligence in medicine is not new, it is the widespread emergence of 3 technologies that are driving its use in pathology. These include the digitization of pathology slides, the development of multiplexing technologies, which allow us to more comprehensively describe cell types and cell states, and advancements in artificial intelligence, such as deep learning and foundational models. Let me briefly describe the engine behind Nuclei's spatial biology technology. The first step is to ingest a digitized slide. The slide can be an H&E slide, an immunohistochemically stained slide, or a multiplex immunofluorescence slide. Once ingested, a series of AI models are executed that include a quality control model to identify artifacts, a tissue segmentation model to identify areas of tumor, necrosis, stroma, etc., a cell classification model to identify cell types and states, and models to quantify spatial features such as neighborhoods, immunophenotypes, tertiary lymphoid structures, etc. And of course, the last step is the analysis of data to inform drug development decisions, such as optimizing biomarker scoring, determining biomarker prevalence, predicting response to therapy, predicting genomic status, and better understanding the mechanisms of action and resistance to a drug. So here is an example of an analysis of an H&E slide of a non-small cell lung cancer, showing the basic steps of analysis that I will go through in greater detail in the next few slides. So after the slide is digitized, an artifacts model identifies areas of the slide that should be removed from further analysis. This is essential so that we don't create bad data by assessing areas that should not be assessed. Next, a tissue segmentation model is run that identifies the pertinent areas of the slide. So you can see here highlighted in the different areas, the tumor highlighted in blue and the stromal areas highlighted in the lighter pink color. Then cells are identified and classified based on deep learning models that have been trained in a variety of different ways. First generation models were trained based on pathologist annotation. But as you should know by now, pathologist annotations all suffer from the same interpathologist agreement that we've seen over the past 40 years. While pathologists are fairly consistent in annotating tumor cells, benign cells, and lymphocytes, they are less consistent in annotating myeloid-derived cells like macrophages and dendritic cells. And to reliably identify subclasses of lymphocytes like cytotoxic T cells, regulatory T cells, and memory T cells, annotating and training on data sets capable of reliably classifying those cells, like multiplex immunofluorescence, is necessary. The current cell classification models identify tumor cells, lymphocytes, plasma cells, granulocytes, and fibroblasts. Now, once the cells have been identified, other areas and cells can come together and extract hundreds of different features. You can think of the whole process as a pipeline, which has many similarities to what was developed for next-generation sequencing. Similar to how a capture kit in next-generation sequencing determines what molecular variants can be detected, the input slide determines what areas, cells, and features can be extracted. More complex areas, cell types, and cell states can be reliably extracted from a high-plex immunofluorescence slide than a standard H&E slide, but value can still be extracted from all slide types. Whatever the input, the pipeline will output areas, cells, secondary structures, IHC patterns if applicable, and a wide variety of spatial features like tumor area, cell distributions, and cellular interactions. There are also special pipelines that can perform tasks like virtual multiplexing, in which two single-plex IHC slides can be combined into a virtual duplex stain, or identify lymphoid aggregates and distinguish them from mature and immature tertiary lymphoid structures. One critical advancement is allowing pathologists to interact with slides and configure areas for more intuitive analysis. For example, when we see a tumor nest surrounded by stroma, we can easily deep learn those two areas and reproducibly identify them. But what if we're interested in the interaction of those two areas? We can take the tumor nest and then, in a configurable way, separate it into tumor at the edge of the nest that is of immediate interest to us, abutting the stroma, and tumor in the center of the nest, which is less interesting in terms of interaction with the stroma. Similarly, we can define the stroma as stroma that's adjacent to the tumor and stroma that is remote from the tumor. The margin of the tumor could then be defined as the outer edge of the tumor nest along with the adjacent stroma. In this way, we can define many different areas which help us better visualize biology as depicted in the image in the bottom left. Most importantly, the pipeline enables us to calculate and visualize features that are beyond the scope of our human vision. For example, in immunotherapy, multiple reports have shown an association between tumor-infiltrating lymphocytes and clinical response. Manual assessment of tumor-infiltrating lymphocytes is limited, but with our pipeline, the analysis of spatial features improves the characterization of tumor-infiltrating lymphocytes in the tumor microenvironment by assessing features like tumor-infiltrating lymphocyte fractions and densities, their heterogeneity, and proximity to tumor cells, as well as other features like fibroblast barriers. The use of spatial biology and AI has many applications in the drug development process, from early discovery and translation through clinical trials and commercialization. Nucleai offers full-service support for the full spectrum of drug development. Complex slides like high-plex immunofluorescence or spatial transcriptomic slides allow us to define significantly more cell types and cell states and are optimal for hypothesis generation. For hypothesis testing and other translational activity, lower plex slides, either chromogenic or fluorescent, are frequently used to ensure that key cell types and cell states are captured, while for clinical trial testing, where single biomarkers are well defined, traditional immunohistochemistry and H&E slides are the most frequently used. But as trials and therapies get more complex, we are seeing some interesting trends. For example, there are more than a dozen ADC drugs in development for non-small cell lung cancer, but lung cancer tissue is too limited to test for 15 different targets with traditional immunohistochemistry. Therefore, we're developing a pipeline to assess the presence, distribution, and amount of each potential target from a multiplex immunofluorescent assay currently being developed by Lunaphore. With that as background, I'm now going to turn over the presentation to Justin Lucas, Head of Translational Research at Aldai Nortye, who will discuss how they used our platform to predict clinical response from H&E slides. So Justin, over to you.
Sure. My name is Justin Lucas. As Ken introduced me, I'm the Head of Translational Research at Aldai Nortye. We've been focusing on a molecule that is a pan-PI3 kinase inhibitor in late-stage development. We have been trying to identify unique biomarkers that could identify potential responders to our therapy in head and neck cancer. We partnered with Nucleai to look at a phase 2 trial that was a positive phase 2 trial to identify H&E slides to look for novel biomarkers to continue to progress this therapy. So as Ken very thoroughly described the utilization and the benefits of H&Es compared to some of the other biomarkers that are typically used in clinical trial research, such as sequencing or multiplexing, the H&E is cost-effective. It's readily available in that all patients have a diagnostic slide in order to be diagnosed, graded, and staged. So we don't need to rebiopsy or ask for additional material from the patients on the clinical trial, and heavily underutilized in the clinical setting. So the focus in working with Nucleai, this is a logical biomarker or source of biomarker material to try and develop tests and assays to identify therapeutic response in the clinical setting. Ken kind of went through in his previous slides these different areas and how they're different, how they're being classified by Nucleai, off of the H&E through the AI-driven and pathology-supported mechanisms. So just to remind everyone, because we will touch on some of these points while we are presenting the data, that we have the tumor area, tumor microenvironment, the tumor invasive margin, and the tumor center, and the adjacent tumor microenvironment. So these are just the different regions of the tumor that can be easily identified by the artificial intelligence off of the H&E based on the morphology and the proximity to different types of cells. So one of the first things that you do when you're building one of these predictive models is to understand the reliability or the predictability of this potential model versus a trained pathologist. And so there are two different areas, as Ken described, that we would look at. We would look at the immune cells, and that's the table on the bottom here. You have the lymphocytes, fibroblasts, plasma cells, granulocytes, and endothelial cells. And you have the other cells in the tumor, such as the tumor cells, the stroma, the necrotic areas, the nerve, and then your other classified cells. So as you can see, the model that Nucleai developed off of our H&E slides was highly predictable versus the control pathologist reads for the tumor, stroma, and necrosis and nerve. You have some more variability, but very high levels of correlation between your lymphocytes, your fibroblasts, your plasma cells, and your granulocytes. So the model can identify very easily and reproducibly these types of cells within the tumor microenvironment. And that, again, as Ken described, provides the foundation for us to start to draw conclusions on their relationship to each other and then to therapeutic outcome or other things within the clinical setting. So how do we actually use H&Es in these models to actually look at and benefit our therapeutic models, right? Therapeutic drugs. So the Barrel-1 trial, again, was a phase 2 trial. It was looking at papanosib, which is a pan-PI3 kinase inhibitor in combination with chemotherapy in metastatic head and neck cancer. There had been an analysis done on what we would consider traditional biomarkers, such as sequencing, immunohistochemistry, and tumor mutational burden. All of these biomarkers did find, were able to find correlations with outcome within these different traditional biomarkers, such as specific mutations such as P53 and PIK3CA, which are known to be altered by our therapy, had strong associations with outcome in the clinical trial. We also saw that tumor infiltrating lymphocytes, or TILs, that's a combination, it's a CD3 biomarker, as well as a CD8 biomarker, which just identifies the active or the killing immune cells versus the CD4 helper immune cells, both also associated with outcome in the therapeutic setting. We also found that high tumor mutation burden, which is basically an indicator that there's some sort of immune component, showed a strong outcome with therapy. So these were the main findings from the original analysis with the traditional biomarkers. So we wanted to understand, if we looked at the H&E and we allowed the algorithm and the computer to provide novel insights based off of the H&E and the morphology of the tumor and what it was showing, what we actually would be able to understand or learn. The first thing we wanted to do before we looked at anything novel was determine, could the H&E slide that we had and the model that we built with Nucleai reproducibly and accurately compete with the traditional biomarkers, or the what we'll call the standard biomarkers, right? So what we first looked at is the tumor infiltrating lymphocytes. This is a very common biomarker, especially used in immuno-oncology. And the 10% cutoff is a pretty standard cutoff. If you have more than 10% of your immune cells that are CD3 positive, you would be in one category. If you have less than 10% of your immune cells being CD3 positive, they would be in another category, right? And so that's how we have to always set in biomarkers some sort of median or divide between the two different categories that we're comparing with clinical outcome. So as you can see on the left, this is the result from the H&E analysis, where we looked at tumor infiltrating lymphocytes comparing above and below the 10% threshold. And on the right, we have the traditional IHC analysis. And these results are nearly identical in terms of the patients and the outcome that was predicted off of the H&E versus the additional slide and reagents needed to compose and conduct the IHC analysis. So this was a very good litmus test to say that, yes, the model that we built is not only comparing to the gold standard of the pathologist reading of what these tumors show, which we showed earlier with the correlation, but not only that, we can now find and identify and confirm similar biomarkers that we know would work if you were to use a traditional biomarker. So then it allows us to take the next step and say, what else could we learn from the H&E that you would not have in the traditional biomarkers or easily, readily available in the traditional biomarkers? So one observation that we made was tumor microenvironment heterogeneity. So what that means is there's a high diversity of immune cell types within the tumor microenvironment compared to a low heterogeneity, so a more consistent cell type. And again, to preface, these are all from the archival H&E slides. So what the patient's tumor looked like when they were diagnosed. And when you build these models, this is typically when this application is put into place, as you oftentimes are not rebiopsying your patient for this, or you don't want to have to expose the subjects to another biopsy just to determine this biomarker. So you can look at the archival baseline material and understand how they're going to respond to therapy potentially years down the road. So in this scenario, you can see that patients that had a higher diversity or heterogeneity of immune infiltrating or different types of immune cells within the tumor microenvironment had a significant survival benefit compared to those patients that were below the median in terms of the heterogeneity of the tumor microenvironment. And again, this is a novel observation compared to the ability to look at this effectively or efficiently in a traditional biomarker. To run any kind of multiplexing IHC or sequencing would be much more costly costly in terms of financial costs as well as material on the slide. And you can see the significant hazard ratio improvement of 0.4 versus 0.99. This also helps confirm some of the mechanism of action of our drug, because a pan-PI3 kinase inhibitor acts on a multitude of cell types, and it's not just your it's not just your T cells, it's not just your macrophages. And this kind of was a very nice observation to show that this molecule is working in a way that we believe biologically that it should. We also found another novel observation around granulocytes, which are another cell type that can be challenging or tricky based on immunohistochemistry for the known biomarkers or the known cell antibodies that detect granulocytes. So in this analysis, we looked at the median granulocyte infiltration, and we went above and below. Again, all biomarkers without a baseline need to have a median cutoff to look at high versus low. So that is within this study of the Beryl-1. But we do see that patients that had a higher infiltration of granulocytes performed better on the combination arm than those that that had lower infiltration of granulocytes in the combination arm. So we want to basically bring this together and understand why we want to use spatial biology in the translational setting. And there are many benefits. And as we kind of highlighted, some of those things are the cost, availability, reproducibility, as well as the potential novel insights into new mechanisms of action that can help you understand how your therapy is working, that you may not be able to determine. Also feasibility, if you were to try and do all of these assays and determine all of these different cell types off of just more traditional biomarkers such as IHC, you would need multiple slides, multiple stains, and multiple analysis platforms to analyze this. All of this kind of biomarkers are rolled into one system that is learning as you feed more information into it. So that was a phase 2 trial that we looked at. As we expand into our phase 2 trial and we have our results, we are able to feed the results from that study and have this model grow and learn and understand and become stronger and confirm what we saw previously, to now make the model even more predictive and more accurate as we go to treat patients in the future. The other benefit for these types of platforms in a clinical setting is there can be a multitude of applications outside of just clinical response. As we looked at in this particular study, there's the potential for diagnosis as not with Nucleai, but with Page AI, they were able to get a AI model that actually diagnoses or supplementally diagnoses prostate cancer, approved by the FDA back in 2021. And so in theory, you can think of this platform towards the future being accessible to local doctors, the same as your academic leaders in major cancer centers, that can accurately and consistently, because there's no inner pathology variability, grade and stage a tumor, and then also simultaneously off of the same image give you a strong prediction versus whether or not the patient would respond to a particular therapy. So it gives a very strong, powerful data set for physicians to easily access and utilize when holistically treating the patient from diagnosis all the way through therapy. So this is a very strong platform and technology that we have been using and we plan to continue to use in this field. And we look forward to working with Nucleai in the future as this progresses. Thank you guys for your attention.
Thank you. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. We'd like to ask attendees to take a moment after the webinar has ended to take our exit survey to give us your feedback. Let's begin. The first question is, what features does Nucleai AI use to identify different tissue regions from H&E?
So I should have spent a little bit more time describing AI a little bit. So Nucleai is using deep learning to identify virtually all of its features currently. So that would be both both areas on the slide. So those are areas that I described already, things like tumor areas, stromal areas, areas of necrosis, artifacts, et cetera, benign areas. You can deep learn to any of those things. And what that means is that the system isn't looking for specific features. It has a training set in which a pathologist has annotated those features on the slide. And then that's presented to the model, and the model then derives those things. So the model is as good as the annotation set that is used.
Okay, thank you for that. The next question is, can spatial AI help to identify tumor origin?
Boy, that's got some really interesting questions. So I'm not sure spatial biology by itself can help do that. But the job of a pathologist has always been to classify tumors. And we've traditionally classified those tumors based on morphology. So given that spatial biology is an extension of that morphology, the answer is sure, that's what we do all the time. That's what pathologists actually do. Now, unfortunately in pathology, there's many things that look the same. But as we got more granular with molecular analysis, immunohistochemistry analysis, we recognized that many tumors that look identical to us on an H&E actually have different proteomic or genomic features. And as we've explored those, you can go back and learn the newer insights and how to distinguish those. Sort of like being able to distinguish brother and sister, fraternal twins, and then identical twins. Some are easier, but when you get into the identical twins, you really need a lot of experience to be able to do that. So I think the answer to that is potentially yes, but if you use those technologies today, my bet would be that they would be similar to the pathologists that they were trained against.
Okay, next question. Is Nucleai AI, or is Nucleai completely trained on pathologist annotations, or how's the ground truth generated?
So for different pipelines, the training is different. We started like everybody else to pathologist annotation. And like I said, and as Justin shown, that works really well for things like tumor cells, lymphocytes, granulocytes. But some of the cell types that are harder for pathologists to reproduce miraculously get left out of those observations. So you don't see a lot of deep learning off of H&Es that talk about dendritic cells or NK cells or cells that would be harder for pathologists to reproduce. To really identify those cell types and cell classes, we just use alternative methodologies. Sometimes it's as simple as a singleplex IHC. So maybe for a macrophage, something like a CD68. But sometimes we want to be even more granular than that, and you need either several stains or a series of genomic tests to better identify that cell type and cell state. And so if you deep learn to that, then that becomes your true set. And we've got some experience with that, but that's the emerging technology that we're seeing moving forward. So a ton of experience with that, for example, with multiplexed immunofluorescence, where we have an incredibly strong pipeline. So if you have an MIF panel, our pipeline can dissect those cell types and cell states and give you much better insights than you would get with traditional methodologies.
Thank you.
Yeah, sorry, I just wanted to also kind of chime in on that. The macrophages are going to always be a challenge for any kind of histological basis. Ken said the shape, but also the size. Sometimes they span multiple sections, and so the nucleus may be on a different section than the actual membrane. And so, as Ken indicated before, they're trained based on finding the nucleus and the border of the cells. And so, there is going to be inherent challenges with those larger and more ambiguously shaped cells in any of these types of analyses.
Thank you. Moving on to the next question. Can these tools be used programmatically to detect tissue types and cell types for our H&E? And are these tools able to identify macrophage and dendritic cells with high accuracy?
So like we said, if you are trained off of H&E alone, the answer to that is probably not, or it depends what you call high accuracy. You'll be in the probably 70 to 75% range. So if you consider that high enough accuracy, sure. But you can train them to immunohistochemical stains. So for example, you can train the detection of CD68 positive cells, assume that those are macrophages, and increase that performance relative to an H&E. So the answer is just the model or the type of slides that you're looking at and the training that you want to undergo to really play out that model. I don't know of a robust H&E model today that's generalizable that can reliably distinguish dendritic cells, macrophages, et cetera, on an H&E. They're pretty good, but it depends what quality you require for your particular problem space.
I just also think that macrophages are tough even with IHC, CD68 versus 163, right? I mean, they detect different cell types. There are a bunch of macrophage markers by IHC, so they're a tough cell type, and I know that people are working on them, especially with the immuno space. They are a very interesting cell population, but from a biomarker perspective, they can be very tricky to nail down and accurately and reproducibly study for a lot of the factors that we've just been kind of mentioning.
Absolutely. It's really important in my mind, for non-pathologists, they'll frequently just say, oh, we're finding macrophages, when in fact you're finding a CD68 positive cell or a CD163 positive cell. And so you've got to define very clearly what you train to, because as Justin said, I could train to CD68, I can train to CD163, I can train to, it has to have both CD68 and 163. I can have it, make sure it's missing other markers in there. So you can get much more complex definitions, and it makes it sound like we're all talking about the same cell or cell state, when in fact we're talking about many different ones.
Yeah, and again, macrophages, right? M1 versus M2 are very different cell populations altogether, what they do, right? So these are just inherently tricky cell types, right? And they're, so I don't think it's as simple as can you identify a macrophage, right? So it's a loaded question, but it's a good one. But it's tough to do on any platform, let alone just off of the H&E.
Okay, let's move on. Next question, is Nucleai compatible with other technologies? I guess like Lunaphore and RNAscope. Is it more compatible? I don't know. I'm having trouble reading the question. It's asking about compatibility with other technologies, and Lunaphore plus RNAscope are mentioned. For example, Stellaris RNA fish.
Yep. So we just take slides as input. So we've worked with all of those platforms before. So the answer to that is yes. But technically anything on a slide that we can learn to something, models can be developed. I'd say our mainstay are things like the high-plex immunofluorescent states, because I think spatial biology in its essence really wants to understand as many different cell types, cell states, and areas as it can. An analogy that I like to use with that is, let's just say that we were people, right? And we want to understand, like, is there, some plot going on in our city, right? So we can take our city as a whole, we can break it up into neighborhoods, we can break those up into buildings, we can break those up into floors of buildings, and then we can start analyzing. And that's sort of what we do with tumors. There's a big tumor. Somebody biopsies a piece of the tumor. We section a piece of the biopsy, and then we take a 5-micron section of that and look at it intently. And from that, we try to extrapolate back to what's going at the hole. And what you're going to extract from that slide, you want to be able to extract as many features as you can, as granularly as you can. So for example, let's say I was doing that building and I take a floor of a building and that's all that I can do is do people. And so that's all that I can tell is, are they male or female? That gives me some indication. I might be able to go a little bit more granular and say, well, are they children? Are they adults? Are they seniors? That would give me more granularity. It might allow me to find, for example, a nursery or a school, or find a senior center if I got lucky. But the more granular I get, the more relationships that I can ascertain, and the more predictive I'll probably be about what's going on. If that's all that I needed to know was, am I in a nursery school, for example, then just the age would have been good enough to do that. So it depends on sort of what your question is. What feature sets do I need to appropriately address those questions? We'll take any data set that's out there. And in my mind, the more complex the data set, the better, because it means you can see less of it with your human eye. And it means that I have more cells and cell states to interact with to show you relationships that you probably wouldn't intuitively see. Hope that made sense. I know it was a little bit complicated.
Thank you. The next question we have is, the novelty of cell heterogeneity in TMI is provocative. Can you expand on what cells are scored, i.e., is it immune and stromal cells such as fibroblasts and endothelial cells? Have you tested its predictivity in tumor types other than head and neck?
So the answer to all those questions is yes. So obviously these tools have been used in virtually all of the common types of tumors that you would think of. So certainly things like lung cancer, breast cancer, colon cancer, pancreatic cancer, melanoma, lymphomas, prostate cancer, et cetera. The common cancers, of course, have been tested. The standard cell types are extracted fairly well from H&E slides. So again, tumor cells, a variety of different stromal cells, fibroblasts, endothelial cells, muscle cells, et cetera. So all of those things can be done, but as Justin has alluded to, some of the most interesting cell types are some of the more difficult ones to identify and exist in multiple states. So it's not just like there's a macrophage, and macrophages are very plastic and they express a range of different states. So identifying that full complexity is much more difficult and requires more complex data as input, either multiplex immunofluorescence or some form of spatial transcriptomics, so we can truly identify what's going on with those individual cells.
Yeah, and again, just to highlight for the crowd, right? To Ken's point, the plasticity of a lot of these cell types, no matter what platform you use, you are looking at a snapshot of a fraction of a tumor. So, there is some benefit to getting that level of granular detail on these cell types and understanding what you're seeing in that particular moment that the biopsy was taken. However, there is also a risk of diving so deeply into that piece of information that is a small representation of the larger tumor, also archival if you're looking at a metastatic disease state, right? So trying to pull out mechanistic biology sometimes becomes convoluted. Also, right, these cell types, even your macrophages are a different story, but even your CD8 cells, they change, right? And in an hour or a week after you take that sample, the population, not even the particular cells, but the population could look very different, irrespective of therapy or change, right? So that's the other, in my mind, in my view, the beauty of the H&E is that you're able to efficiently kind of understand what's going on at a level that is sufficient enough to draw therapeutic conclusions without having to dive deep, because if you were to do multiplexing sequencing, that's 6 slides minimum, right? Sequencing is sometimes 10 slides in addition, right? And those materials, you can extract them and sequence them and not even get viable data out of them, right? And so the H&E is a much more reliable and robust source of information when you are trying to put together a biomarker strategy for a clinical trial. And if you can make correlations and conclusions based off of that, it is a much more robust platform, in my opinion.
Those are great points, Justin. And, I'd like to point out, to expand on that, relative to heterogeneity in many tumor excisions, as pathologists, we look at multiple different areas of the tumor on an H&E, but we select just one area for biomarker analysis. And by being able to execute across all of the different H&E slides that we have really allows us a much better assessment of the heterogeneity. And there's great studies on deep molecular analysis that show that when we identify mutations in tumors, if we had the breadth of the tumor there, the tumor and its metastasis, et cetera, that only about a third of the mutations that we identify are actually ubiquitous, meaning in all areas of the tumor that we looked at. So being able to extrapolate from H&E slides to better predict areas of tumor for deeper analysis becomes really important as well.
Great, thank you. We'll move on to the next question. Does Nucleai, I just asked that question. How do you imagine the path to clinical utilization of such technologies will look? Regulation, reimbursement, workflow integration, et cetera.
So those are fantastic questions. I'd say that's the $64 million question, right? Is how do we move these things into the clinic? And I like the thought process that the AMA has sort of laid out for artificial intelligence in general. So there's tools that exist today, and I showed you some of those, that I would call AI assist, right? The pathologist does its job. It scores a HER2 slide. It makes a diagnosis of prostate cancer. It does whatever it's going to do. It assists the pathologist. And those are absolutely fantastic because they harmonize us. They democratize the interpretation. They prevent us from overlooking things that we might have overlooked. But you have to remember that the tools aren't perfect, and sometimes the tools are mistrained or pick up something that they shouldn't have picked. So those are assistive. You still need the pathologist or the clinician in place. And the problem with those tools aren't that they're necessary, because those tools are fantastic and have been around for 20 years, and pathologists love to use them. The problem is nobody pays for them because they're QA. So payers look at that as that's an quality assurance, that's on you. And so if you want to pay for that, go ahead. So the two barriers to entry to that are, one, you have to digitize all your slides, be fully digital, so you can do that. And second, there's no real reimbursement tied to it. The second level, which I think we're all excited about, is what we would call AI augmented. And that means the pathologist does what the pathologist does, but the tool does additional things that my eyeballs are incapable of doing. It actually gives me insight beyond what I could possibly do on my own. And I think that's the wave of tool that we're waiting for next. It's a big risk for pharma to go there because they would have to get regulatory approval on that. And it's a high bar for labs to prove that whatever they're developing is actually additive to what they're already doing. But I think that's the next wave that we're going to see. We might be a couple years away from that yet. And I think we're going to see reimbursement tied to that, right? That's where you're augmenting what a pathologist can do. And then, of course, the last, is the omnipotent AI. You just give the slide to the AI and the AI tells you the answer and you don't need a pathologist there. I think we're many, many, many years away, decades away from that. By the way, I think that's achievable at some point, but with tens of millions of training slides in with known outcomes to get that level of prediction. So I think it's just time. And the first barrier is going to be, for pathology at least, is widespread adoption of digital pathology, which is, has had some major milestones over the last decade, but it's still probably only penetrated in maybe 15% of the pathology world.
Yeah, that's really well said, Ken. And again, that I think that last point is something that we want to, I think we should really make sure is driven home, right? The training and the utilization can only really benefit, right? Companies, big pharma, small pharma, biotech, we can all do these experiments and generate really cool data. Without the widespread adoption of the platform, there will not be a mechanism to move this forward, right? We had the first AI augmented or assist, right? I forget, I don't know how you classify the Path AI, diagnostic. I think it's kind of in between those two. I guess it's an assist, right? It's not really augmenting because they can do that because it's efficient. But we had that approval, right? The FDA approved that. It's a test that can be ordered, but there's no adoption because the doctors and the physicians who need to use that or would benefit from that don't even have a scanner, right? They can't even scan the slides and use that. So the platform, the infrastructure really needs to be there before we see traction with the regulatory bodies and things like that, right? We're putting the cart before the horse, thinking about the FDA. We need to get the doctors on board and the pathologists on board kind of across the field before you see any kind of real regulatory traction, in my opinion.
Thank you. Next question. Does Nucleai AI work with other diseases or normal tissues in addition to tumor tissue?
So we focus on two general areas. So one is tumor, and we've got experience, obviously, with all of the common tumor types. I'd say the 15 most common tumor types for sure. If it's going to be a model of something that's incredibly rare and doesn't translate well to other tumors, sort of like a one-off, like maybe a rare childhood malignancy, there'd probably be additional training that would be necessary to identify reliably that particular tumor type. And what was the second part of the question? There's another part.
Was disease tissue or normal tissue?
Yeah. So the second area, so tumor is one area that's most of our concentration because we're focused on pharma, and most of their questions have been around antibody drug conjugates and immunotherapy, as I sort of described. But another area that goes along with immunotherapy that's not tumor is inflammation. And so things like inflammatory bowel disease, eczema, rheumatoid arthritis, all of those autoimmune type diseases have the same spatial need of understanding what the inflammatory infiltrate looks like, how it's arranged, how it distorts normal cells. And so, yes, we have models for those as well.
Okay, we have time maybe for one more question. Is there any accounting for staining method or reagent differences as it applies to the H&E samples? What about differences in imaging modality or instrument?
So another fantastic question. All of those things make a difference, potentially. So what we care about when we generate these H&E models, I spend a lot of time talking about, well, maybe we need to do better than pathologist annotation, which is a true statement. The other thing that you have to take into consideration is the variability in tissue. So different thickness of the tissue. Not all labs cut sections at the same thickness. They don't all use the same paraffin. They all have slightly different H&E stains. Some are redder, some are bluer, et cetera. And then the scanners aren't all the same, right? Some have different objectives. They might say, oh, I'm scanning at 20x, but the numerical aperture of the objectives, the point-to-point resolution, the brightness can all vary significantly. So we overcome that in our training models. We use a lot of techniques to alter the stains that we've got to where we can virtually alter the stain to get us more generalizability. But that's a question that you want to ask of your AI models is how generalizable is it? And it really means what's the robustness of your training? Have you trained to the full extent of tissue that you might see? So we've done that and published on that for things like different PDL1 clones run on different instruments, scanned with different scanners, and shown the robustness of that between stains, for example, like SP263 and a 22C3. So it is something that's incumbent on that AI lab in defining the model to make sure that they've captured the full spectrum that you're interested in being able to, that you might see in the real world.
Thank you. That's all the time we have for today. We'd like to thank Justin Lucas, Ken Bloom, and our sponsor, Nucleai. If we didn't have time to get to your question, we'll try to follow up with our experts. As a reminder, please look out for the survey after you log out to provide your feedback. If you missed any part of this webinar or would like to listen to it again, an archived version will be emailed to all attendees. Thank you for joining us for this Precision Medicine Online webinar.